the last lecture was going over toxicology and then some of the carcinogens in the topic of toxicology, but now we're going to talk about risk assessment, which is like an overall framework of how to frame contaminants, environmental contaminants, and how they interact with our bodies and our lives. So I'm starting this off by showing you some of the EPA and CDC websites. Now, everything that we interact with here in the U.S., a lot of things that we buy every day, makeup or food, supplements sometimes, even pipes for our water and our toilets, all of that is approved and based on certain standards. And those standards are based on science and a process for approving those things. And that is environmental health risk assessment. That's the framework for evaluating everything and also to determine if something possesses the potential capacity for a health risk or a detrimental health effect. So I've got a couple of websites here on this title slide. The first one is the EPA risk assessment and what that is, and then I have the IRIS and the ATSDR. And these are two sites. One is from the EPA, the IRIS, and this one, the ATSDR, is a CDC website. And they both have many contaminants and all the different types of studies that have gone over those contaminants and all the animal studies, and they quantify the risk for according to the current known science using many peer-reviewed articles and many other sources. So those are two websites that sort of bring everything together for you. This first one here is sort of a history of what risk assessment is at the EPA. They were first, this website here, it goes through what risk assessment is. In December 1975, they had the quantitative risk assessment for community exposure to vinyl chloride. That was sort of the first risk assessment that was synthesized and done and explained via this format. In the 1980s, things got a little bit better with water quality, and then in 1983, the National Academy of Science had the Red Book here, and that is the one where they go through each one of the different known contaminants where there's data, and they develop a framework there for which to evaluate all of the different types of contaminants. Another website I have on this page here is this right here, the IRIS. And so that's the IRIS is the US EPA's method to contain all of the different topics on the different chemicals that are out there. So you can go to the IRIS or the ATSDR. So say you're interested in vinyl chloride, where you can click here on the IRIS and you can look at what is the health risk of that particular chemical. So I have this figure here to sort of explain what the risk assessment framework is. So it starts out with hazard ID, exposure assessment, and dose response assessment, and then risk characterization. And it broadens out into risk management and risk communication. Now, these are broad circles that include a lot of different topics, and all of this is all public health. But I'm going to talk about this blue one here, which is the hazard ID to exposure assessment, dose response, and risk characterization. Now, with these lectures, what I do is I split up risk assessment into those four categories. Hazard identification is sort of the first occurrence of the community or the individual noticing that there's a hazard, or the scientist, the researcher, somebody quantifying there is actually a hazard and going out there and actually talking about what that is and doing something about it. Then it moves on to the two next stages are exposure assessment and dose response assessment. Exposure assessment is what happens outside of the body, and dose response is what happens once the dose of whatever contaminant that you're exposed to is inside the body. So this could be an acute effect or a carcinogenic effect. So it could be something that develops a symptom in your body over a long time, and it could be a cancer, or maybe it's just something that develops over a long time, which is a poor health effect and not necessarily a cancer, something like emphysema. Or it could be a short acute effect, so that would be something that you develop quickly. Say you develop some neuropathy from being exposed to a high dose of pesticides, something like that. or even a burn, a chemical burn is an acute poor health effect. So those are the hazard. That's the hazard ID. You first go, you recognize that something's a contaminant, and then you move on to dose response assessment. You can also move on to exposure assessment, and dose response and exposure assessment sort of start at the same time. Exposure assessment looks at how things move through the environment and where it goes, where it ends up, and how it eventually gets to you. So what you do for risk assessment is you make estimates of what is your risk. What is the risk of getting sick from or having an ill health effect from being exposed to any particular contaminant. So I'm starting here with hazard ID, and again, there's hazard ID up here in the corner, exposure assessment, dose response, and risk characterization. How does the hazard start? What are the chemicals of concern? What is the description of the chemicals, and why are the people or the person concerned about this chemical? Do they think there's some kind of unique exposure that they have? And how does the actual hazard exposure start? So you're just answering some of those basic questions in hazard assessment. You know, you do a lot of different things with this. You can do community surveys, ground truthing, crowdsourcing, needs assessments, windshield surveys, environmental impact assessments, health impact assessments. You know, there's a lot of these basic first impressions sorts of qualitative methods are used for hazard ID. So you could drive around San Bernardino and look at all of the water wells that have a filter on them, and then you could say, well, what is the hazard here in the water? You know, why do they have to filter all of this? And then you do your research and homework and figure out what the contaminants are. Or you could go and drive into a disadvantaged community and just look around and see that maybe there's a lot of people there that work with pesticides, and they have pesticide trucks just parked all over the community, and some of them are leaking, you know, a product onto the ground, for example. You know, that's another type of thing. You're doing hazard identification. The next step after that is actually quantify. You want to ask, well, it's leaking something, but just because it's leaking, does that mean that anybody's at risk? Maybe it's just leaking water. Maybe it's a water tank, and you just assume it's a pesticide tank. So there's a lot of things you don't know, and the process and framework of risk assessment allows you to start answering those questions. So factors that affect the concentration and toxicity of a chemical are also sort of quantified in or the hazard ID. So you can start hypothesizing of these things, but really these factors are more exposure assessment. They kind of cross the line a little bit. So how long are you exposed? The duration of exposure. So who received the dose of the chemical, and how did it get into your body? What is the route of entry? And then there's also things to consider. Individuals have different sensitivities, and there's also interactions of different chemicals that'll do different things. So the routes of exposure: ingestion, injection, contact, inhalation. So those are 4 routes of exposure. There's really 3 that are important here: ingestion, contact with the surface of the skin, so like dermal or topical, and inhalation. Injections is really like a healthcare concern, or for people that use needles every day. There's certain contaminants to be aware of with that. But really, the 3 ones that we're talking about are those 3 for humans: ingestion of food or water, contact with the skin, or inhalation, you're breathing it in. So really, that's a lot of different possibilities there. Injections into the bloodstream is something that happens in healthcare. There are other methods as well, other routes of exposure. One of them is when you're doing rat studies or rodent studies, you sometimes will introduce something directly into their stomach by gavage with a big needle. So you stick a needle down their throat and then introduce the contaminant in there to see what happens with the rodent bioassay. Another one is the type of bioassay is, you know, you can rabbits have eyes that are more similar to human eyes, I guess. So they use a lot of rabbits to test some cosmetics and makeup products and things on eyes. So you'll do some studies with them. So that eyes are another type of route of exposure. But for us, it's the ingestion, contact, and inhalation. So there's all of those routes of exposure, and that's to quantify exposure assessment. So exposure assessment is really to characterize the setting, the physical environment, the potentially exposed populations. You identify the possible pathways, where was it released, how long were people exposed for, what is the source of release, and then you quantify the exposure. How many gallons of this did somebody drink over the last 2 days, or how many milliliters did they drink, and then what is the concentration of the contaminant in those milliliters of contaminant that person drank.